Good afternoon, everyone. We're so glad that you are here for our EMSL Learn webinar today on TerraForms. We wanted to let you know we'll be taking a break in December and our series will continue in January. We also want to make sure that you keep in touch with things that are happening at EMSL. And the best way to do that is to follow us on our social media channels and also to subscribe to our newsletter. We have a newsletter that comes out generally once a month, and that's the best way to keep in touch with us. So if you haven't already, the links will be posted in the chat, and we invite you to follow along. But today we are here to talk about TerraForms. We have two presenters for you, Arunima Bhattacharjee and Jayde Aufrecht will be introducing a new capability that is available here at EMSL that they developed called TerraForms, Tunable Environments for Exploring Soil Processes. Jayde will begin this webinar by presenting two technologies for zooming into carbon cycling hotspots underground, namely synthetic soil aggregates and rhizochips. Then Arunima will be discussing how you can use pore-scale micromodels, minerals, rhizochips, and SubTap microbial and rhizosphere platforms to look at the influence of carbon distribution on microbe and plant development. There will be time at the end of today's webinar for you to ask questions. We encourage you to post your questions in the Zoom chat so that Jayde and Arunima can answer them during the Q&A. And with that, we will begin with Jayde.
Thanks, Genoa. All right, my name is Jayde. I'm a biologist here at EMSL, and I'll be kicking off the first part of our webinar today on our new capability called TerraForms. These are tunable environments for exploring soil processes. And so we'll be going through a few examples of these TerraForms and how you can use them for your own science. First, I want to acknowledge up front all of the people who have helped make TerraForms a real capability at EMSL. This includes many of our colleagues who have helped generate proof of concept data, as well as collaborators from other institutes who have been early adopters and have volunteered to use TerraForms for their own research. We'd also like to thank the EMSL Science and Tech Advisory Committee for helping us refine the TerraForms idea, and also thank you to Dr. Paul Baer for encouraging us to think about how best to communicate the TerraForms to the user community. Arunima and I are really grateful for all the help that we've had in getting this off the ground. Our primary motivation for creating the TerraForms is that real soil is really too complex. But don't get me wrong, this complexity is very important in orchestrating interactions underground. For example, just the spatial structure of soil alone can affect certain nutrient diffusion, the sparse distribution of resources underground, and those kind of things are going to end up influencing microbial functions. So things like competition between microbes, niche specialization, how well they work together and compete for resources. Those microbial functions, in turn, are going to collectively come together to form some of these emergent ecosystem properties. So things like altered carbon use efficiency, the persistence of some strains underground for longer than others, or even things like pathogen emergence. Unfortunately, though, we don't really have a good way of predicting these emergent ecosystem properties from soil parameters themselves. And because every soil sample is different, even if we sample a lot of different soils, that's really good for helping us understand the correlations between different parameters and ecosystem properties. It's not necessarily a cause and effect sort of relationship. And so that's where our TerraForms are stepping in to fill that gap and really help us understand the underlying mechanism behind some of these soil processes. So TerraForms are tunable. That is one of their major strengths. So if you have a hypothesis, something that you're interested in studying in the field, and you want to test if that's like a real relationship, you can tune the parameters of the TerraForms to really understand what's affecting that process. So some of the soil parameters that we can add back to our TerraForms platform include physical structure of soil, so things like soil aggregate morphology, soil porosity. We can also tune the chemistry to make it more similar to soil. So adding back things like minerals. We can choose the biology as well. So we can culture plants in here. We can also do microorganisms like bacteria and fungi and some of their predators. Then we can add things for the plant's bacteria to eat, things like organic matter and different nutrients as well. So all this depends on the question that you want to answer. We're very flexible. We can make the TerraForms at EMSL and use them here, or we can ship them to you and you can work with them at your home institute as well. So I mentioned TerraForms are tunable, that's their strength, and they can be customized, but we also have what I'm calling the TerraForms Classic. So these are 4 platforms that we've used a bunch and vetted. And so we will be presenting on some of these today, just results that we've gotten as sort of case study, so you can understand what kind of research you can do with these. So we're going to be presenting on synthetic soil aggregates, the RhizoChip, pore-scale micromodels, and the SubTap microbial and rhizosphere platform. So I'll kick us off by starting to talk about the synthetic soil aggregates. We wanted to create a physical model for soil aggregates specifically because they're a really interesting unit of soil. Some research has shown that in each individual soil aggregate, which is less than 2 millimeters in size, they can have their own distinct microbial community. So even though they're very small units of soil, they are still very heterogeneous and interesting to look at. So we are using bioprinting to create synthetic soil aggregates. The synthetic soil aggregates themselves are made of a hydrogel called gelatin methacrylate. They look very unimpressive by eye, just these little blobs here, but when we look at them with an electron microscope, we can see how that hydrogel is helping us recreate the porous structure that occurs within an aggregate. So there's lots of little crevices here where microbes can hide out and form their own niche. And when we characterize this pore distribution in our synthetic aggregates and compare it to what we know about real soil aggregates from XCT, there's quite a bit of overlap there. Making synthetic soil aggregates is a pretty straightforward process. So you can send us the microbes that you're interested in studying. Usually we grow them up in liquid culture and then we mix them with our hydrogel. So the microbes themselves are actually embedded in that hydrogel. The next step is deciding what you want to feed the microbes. So we mix in some solid phase substrates that we want to understand how the microbes are breaking those down maybe. So far we've tested things like chitin, different minerals. We've tried plant litter and microplastics. There's probably other ones we can add if there's anything else that you're interested in looking at. So once you've mixed all those things together, the next step is just to print these aggregates. And once we've printed them, there's several things we can do downstream to analyze the synthetic aggregates to understand what's going on inside. Some of these characterization methods are dynamic, meaning we can look at them over time. So for example, in this sort of proof of concept study, we were looking at mixing together cultures of E. coli and Pseudomonas fluorescens inside the synthetic aggregate and feeding them chitin, which is an abundant biopolymer in soil. And what we saw was that the bacteria had a different multicellular morphology when they were cultured in this structured synthetic soil aggregate compared to just liquid culture. So in the structured habitat, they grew up to form these microcolonies, whereas in liquid culture, they were more planktonic cells. They also had a different spatial distribution, so more closely related when they were cultured together in the synthetic habitat versus liquid culture. So it's cluing us in that the structured habitat is affecting how these microbes are interacting together. Next, we looked at what the microbes were functionally doing inside these aggregates. And so for this, we looked at the ability of the Pseudomonas strain to degrade chitin or produce the chitinase enzyme. So we actually bioprinted into a 96-well plate so we could look at this in more high throughput. What we saw was that in the synthetic aggregates, the Pseudomonas fluorescens actually produced more chitinase than when it was cultured in liquid. And this is just an example. We've done this with other enzymes as well. So we've looked at cellulase and peroxidase as well with at least 11 other strains, I think, so far. And sort of the trend we're starting to see is that bacteria tend to produce more enzyme in the structured habitat whereas hyphenating strains tend to produce more in the liquid culture. And so no solid conclusions yet, but it is important to note that the structure of the habitat can influence function, and so that's something to keep in mind for your experiments. So this is sort of an example of we're moving more towards working with larger communities of microbes in these systems, and not just model organisms like E. coli and Pseudomonas. We're actually working with some soil bacteria. We've cultured up to 11 member communities in these aggregates. And what I'm showing here is actually just pairwise studies between two individual strains at a time, and looking at how those interactions will affect the integrity or the production of the cellulase enzyme. So one interesting thing that we saw was that our fungal strain, Trichoderma, actually had a very different interaction or ability to degrade cellulose when it was cultured with different members of the bacterial community. So we can start to kind of use this platform to understand how microbes interact together and how they're degrading or cycling certain carbon compounds. So once we've done our dynamic characterization of synthetic aggregates, we can also do some endpoint measurements with multi-omics. So at this point, you can choose to send your synthetic aggregate for DNA and RNA extraction to understand more about what's going on genetically. We've demonstrated we can get good quality DNA or at least 16S amplification. And we've also been able to get RNA, high quality RNA from an 11-member mixed culture and within just a single synthetic soil aggregate. So that's about 2 millimeters in size. So it's a pretty small sample. The other option is you can send your synthetic aggregates for a workflow called MPLEX here at EMSL, which is where you can get simultaneous metabolomics, proteomics, and lipidomics stable data from the same sample. So just as sort of a comparison, when you are working with real soil, we really need about at least 4 grams of real soil to run metabolomics on it. And that's because we have to do several cleanup steps so that we don't ruin the mass spectrometer when we put the soil in there. Whereas for the synthetic soil aggregates, because they are much less complex, we can actually do this on a single synthetic aggregate, which is 2 millimeters in diameter. So I don't know about you, but when I see this real 4 grams of soil, I'm thinking to myself, the bacteria over on the left side are probably not interacting with the bacteria over on the right side. And so being able to zoom in and look at the microbes at the microbial scale is going to help us better understand how they are working together and cycling different compounds on that sort of microscale hotspot level. This is just proof of concept data showing the kind of results we can get from the metabolomics, proteomics, and lipidomics on these kind of samples. I'll draw your attention to the lipidomics results. So for example, in a rich broth, if we culture E. coli by itself, we can get about 100 lipid IDs. When we did that in the synthetic aggregate and added only chitin as a nutrient source, we were able to see 42 different lipids. So that is pretty good, I think. Okay, so at this point, I'm going to shift gears a little bit and talk about the RhizoChip platform, which is another one of our TerraForms platforms that's really good for studying rhizosphere interactions and being able to culture the plant. So this is made slightly differently than the 3D printed aggregates. So I'll just kind of walk you through what we do to make this. We actually make these in a clean room by depositing and developing different layers of photoresist on top of each other. So you can think of this sort of like how people used to develop pictures in a red room before we had digital photography. So these microfluidic based terraforms are very customizable. We can get down to like a single micron resolution. And so you can really have a lot of control over changing the design for the specific science question that you're after. The other major benefit is that these designs are they're reproducible. So it's exactly the same for every replicate. It's going to have the exact same design orientation. So that means that we have a lot of tractability and we can actually use simulations to look at what kind of physics are going on inside of these terraforms. So, things like mass transport and diffusion. So this is the RhizoChip. It was developed in collaboration with Oak Ridge National Lab and their Center for Nanophase Materials Sciences. So as you can see, it's transparent. It's large enough to hold a plant and small enough to fit on a microscope stage. The inside of the rhizochip has a soil-like structure that was designed to replicate the sand or the grain size and distribution of sand from a beach in Hawaii. We can grow several different kinds of plants in the rhizochip. So we've tested different grass species like Brachypodium, tall wheatgrass, sorghum. We've tested other plants like poplar and hemp, for example. Most of these plants we can grow in the rhizochip for about a month before they start to get a little bit too big for the rhizochip. But it turns out that the first month of the plant's development is actually a critical time for looking at the establishment of the root microbiome. And so that's where the rhizochip can really help with your research. So here is some examples of optical images we can get with the rhizochip. On the left, we can actually get down to organelle scale resolution. And on the right is an example of how we've used this to probe plant microbe interactions. So here we were growing a poplar cutting in the rhizochip, and we inoculated with a special bacteria that we had engineered to fluoresce green whenever it was expressing a gene for nitrogen fixation. So what we actually saw was that this bacteria would light up green inside the root tissue, which is shown by these blue cell boundaries here. And so that's telling us that these bacteria are fixing nitrogen inside the plant. And we actually were able to identify a potentially novel physical interaction with the plant. So in panel B on the bottom right, you can see these spherical structures inside the root epidermis that we think is a way that the plant and microbe are interacting in order to enhance nitrogen fixation. This is just sort of an example of the kind of images that you can get if you're interested in studying plant microbe interactions with the rhizochip. We can also get several kinds of chemical images to understand the molecular environment that's being created in the rhizochip. So this is some examples of, we can do different chemical imaging. We can do MALDI mass spec imaging, nanodeci imaging. This is a few examples of MALDI chemical imaging here, where you can see how certain compounds like fatty acids, like this palmitate, stick very closely to where the root structure was in the rhizochip, whereas other smaller polar-like compounds like glycolate here tend to migrate further away from the root and can even pool in hot spots away from the plant. These chemical images are really helping us to understand sort of the spatial extent that the plant has influence over in the rhizosphere. And we can actually get many different kinds of primary, secondary metabolites using this method. And so we can image a lot of different compounds. If you are interested in a specific compound, it's likely we'll be able to pick it up. But if you're also just interested to do an untargeted analysis to see what you can see, this is also a great method for that. So as you can imagine, this is a ton of data. Each one of these pixels is about 20 to 100 microns. So it's really high resolution spatial information, lots of metabolites here. So we've been working with Dr. Peter Zor at Berkeley Lab and a part of Camera to sort of try to reduce this data dimensionality and understand the correlation between different metabolites and organisms, and also using machine learning methods, and also to help make the mass spec data analysis more and acquisition more efficient so that we can get more samples through. So one other thing we can do, a few other analytical techniques with the rhizochip. We can do things like nanosims. So if you're interested, like for example, in tracking 13 CO2 being fixed by the plant, exuded and then consumed by microbes, we can actually track that 13C using nanosims. You could also look at, for example, 15N2 being fixed by a microbe and then secreted and given to the plant roots. So this would be a great platform for kind of looking at that spatial distribution of stable isotopes. We can also do laser capture microdissection. So for this, you can go into an individual pore in the rhizochip, cut out your bacteria and send that little piece for RNA extraction or DNA extraction. So you can do sequencing on that. So this would be great if you were, for example, interested in looking at how microbial gene expression varies with different metabolites, spatially, or with distance from the root or something like that. Okay, so the rhizochip is also able to be customized to make it more chemically similar to soil. So you can add different minerals and organic compounds to the rhizochip as well. This is something that my colleague Arunima has been really working on. And so at this point, I'm going to give the presentation over to Arunima so that she can present the final two TerraForms classics. Arunima?
Thank you, Jade, for the nice introduction to the first two platforms. So next, I'm going to describe to you two other platforms. One of them is the pore-scale micromodel, and the other one is the SubTap platforms, which are compatible with both plants and microbial culturing. Okay, so Jade gave a really nice introduction about how we can customize these platforms for adding different types of minerals, and also customize them for like mimicking the soil aggregate structure, porosity, etc. So here is in this platform, which is really called the pore-scale micromodel platform, I'm going to describe to you what we can do when we add minerals to it. So apart from just studying microbial growth inside of this platform or plant microbe interactions, we can specifically look at how plants and microbes interact with minerals. And that's important because we want to look at how certain inorganic nutrients cycle and how they cycle in relationship to carbon in soils. So here, a great example that I'm going to use here is fungus, because fungi are really ubiquitous in soil and they help translocate resources. It could be inorganic nutrients, it could be water in case of drought. So they really provide these nutrients to host plants or even to bacteria when need arises. And so similarly, fungi are really great at degrading minerals. And this is an example of a scanning electron microscope image where, like, if you add minerals to the soil, you can see these kind of like tunneling features that the fungi makes on the surface of the minerals, and they can create certain precipitates as well. So one of our old papers that we studied was to look at how fungi bridges carbon hotspots. So this image that you see here, like the green bright images here, these are chitin that we added to a microscope slide, and then we pressed that against the soil inside of these soil boxes, and we stained it. We observed that fungi was bridging these hotspots, and so they create these mycelial bridges, which are great at translocating nutrients. That you can see this fiber-like, bridges that the fungal hyphae is creating. And so in soil, these processes are really hard to study, and that's the whole point of TerraForms, because these are reduced in complexity. So if you observe a certain hypothesis in soil and how complex soil is, and you want to really test that hypothesis using specific parameters and mimicking those parameters, TerraForms are great. And specifically for microbial interactions, pore-scale micromodels are really great because they're small in size, and you can study these microbial interactions. You can image them. Like in this case, what we did was to create these two carbon hotspots that you can see labeled as C. I don't know if you can see the laser pointer, but basically the green C labeled are the carbon hotspots. And then what we did was inoculating the C1 hotspot with a fungal strain isolated from Kansas soil, and it's a fusarium strain. So in between this carbon hotspot, there was nothing, and the fungus started growing inside of this micromodel device in air. So just to describe this micromodel device, it has like these posts coming off of the surface that you can see, like the dark colored circular shapes. These are posts coming out of the surface, and they're very homogeneously distributed. And so the fungus is growing in between the posts, which is mimicking the soil porosity. And so it grows inside of these pores. And what we observed is the fungus bridged the C1 to C2 only when minerals are present. This kind of mimics a situation in soil where there is a lot of drought happening, and you have these minerals distributed in there where there is not much bioavailable nutrients, but the fungus is able to degrade these minerals and extract it as it grows to another hotspot. So foraging behavior of fungus is usually seen in soil, where they will bridge these carbon hotspots. And this is something that we are able to observe here. We also looked at hyphal density. We can do those kind of things like imaging and quantifying the amount of biomasses inside of these devices. And all of our TerraForms platforms are, the microfluidic TerraForms platforms are compatible with different kinds of chemical imaging. So in this case, you are able to see a time of flight secondary ion mass spectrometry image of potassium being translocated through the hyphae. So in the fungus, as the fungus was growing from two different hotspots, it was extracting potassium nutrient and growing through the porous space to the other hotspot. And then we looked at what kind of metabolite distribution was present around the fungal growth on these devices. And all of our devices are also compatible with different kinds of mass spectrometry imaging. What you saw before in the previous slide here is a secondary ion mass spectrometry image, and this one is a MALDI image, which Jayde also showed you. Here we are looking at what kind of acids, small molecular organic acids, the fungus is producing. For example, it makes tartaric acid, malic acid, citric acid, fumaric acid. The reason for looking at these acids in this study was because fungi, as well as plants, makes these small molecular organic acids to degrade minerals or weather minerals. These acids are also used for uptake of inorganic nutrients. So in this study, we focused on potassium minerals. And so these acids were produced not only to weather the mineral surface, but also to uptake the potassium nutrient. So as I mentioned, these are, again, reduced complexity platforms. So how can you really arrive at that conclusion that it used these acids to uptake nutrients? So for that, we collaborated with SSRL, which is a synchrotron source where we can do X-ray fluorescence imaging, and we can do some spectroscopy to understand the potassium chemistry. And so we looked at the potassium chemistry inside of the fungal hyphae, and we observed that these fungi produces, for example, citric acid, which we saw this fungus make in the previous slide, also tartaric acid. We observed different hotspots of these acids inside of the fungal hyphae. So for example, these red hotspots that you see on the fungal hyphae, these are potassium citrate. We also observed other types of potassium chemistry, like the one in the green, and these are all false colored. We call them organic K. We don't know what is the nature of that potassium chemistry, but we are in the process of trying to figure that out using different kinds of simulations and DFT techniques. And so using different standards, we can compare what chemistry, what kind of potassium chemistry is inside of the hyphae. So this kind of similar technique you can also perform on a plant root, for example, and try to look at what kind of mineral chemistry exists inside of a particular type of biomass. So in this case, we arrived at the conclusion that these organic acids were actually used to weather the mineral surface and chelate and uptake the potassium from the potassium feldspar interfaces, which was on the surface of the pore-scale micromodel. And even the rhizochip that Jayde just presented to you guys are compatible with these synchrotron-based imaging techniques, as well as the spectroscopy techniques. And so here you can see a rhizochip, where there are minerals embedded. In this case, potassium feldspar minerals were embedded. We grew a root inside of this rhizochip, and we looked at potassium chemistry of the root. So in this case, you're seeing a root here that is growing inside of the channels of the rhizochip. There's like the total sulfur map and total potassium map. So interestingly, we can also map all these total elemental maps inside of the biomass. So that's one thing that we can get from these biomass when we collaborate with SSRL. This is also available to users. We have sent several user samples, which was processed at EMSL and to SSRL for further processing. So here again, looking more closely at the root structure, we can see different potassium chemistries present inside of the root. And in this way, we can observe what kind of compounds the root is uptaking and what is the chemistry of those compounds around the root. So this is an example of how you can reproduce different mineralogy inside of these chips and look at interaction between a plant root or a microbe. We can also do this with bacteria if you have a particular bacteria of choice. In general, reproducing these different types of chemistries and porosity aggregate morphology does have it is still a reduced environment, but you try to go closer and closer to soil. And again, like if you have a hypothesis that you couldn't test in soil because of the heterogeneity and opacity of soil, this is the perfect platform for that, where you can visualize these chemistries using optical microscopy and different chemical microscopy methods. Okay, now I'm going to switch gears a little bit and move on to the last platform that we are going to show you today, which is the SubTap platforms. So 2 examples again. I'm going to speak very briefly about the SubTap microbial platform. This was developed by a user at EMSL, and we use this technology to create platforms which are now we are able to use for plant growth. So in this case, you have the SubTap platform is 3D printed, and so what you're seeing here is the blue plate on top is the culture plate, where you can culture microbes, and the gray plate is for capture or capturing the metabolites. So this is the cross section here, where you can add agar and then grow the microbe on top of the agar. There is a membrane separating this the blue plate and the gray plate, and then the metabolites diffuse through the agar and get captured at the agar pad here of the capture plate. And then this capture plate is then taken to for mass spec analysis by a robot. This is extremely high throughput. It is in the configuration of a 96 well plate. And the interesting thing is that you can print these in any kind of configuration. If you want to observe indirect contact of microbes, so if the microbe is basically communicating, two different microbial species are communicating to each other through the membrane without being in contact. So here, this is a configuration that was printed with like 3 different wells, where the microbes are basically communicating with metabolites. So you can do all kinds of studies like that. And since this is printed in the format of 96 wells, you can have an array of conditions. Okay, so that was the microbial platform, but we also have extensively used this for looking at plant microbe interactions. So this is the top plate where we do culture or incubation of the plant. We can grow the plant here, or we can even incubate the plant here and look at metabolites. And this is this basically fits to the bottom capture plate here like a Lego, and they kind of snap together. And you can see the wells here below that is separated by a membrane, and the plant root kind of grows on the membrane. And this is the cross section again, and the yellow agar pad basically captures all the metabolites. This study that we did is also spatial in nature, because there are wells along the root length, which gives you an idea of what metabolite was produced in what section. The resolution is not as great as the microfluidic platforms, but you do this in an extremely high throughput manner, collecting these metabolites, so you do lose resolution a little bit. Okay, so this is how the platform looks like. Here we basically printed several wells where you can see we can grow these plants on agar as well as soil, and the blue plate on the right here is for capture of those metabolites. We do have differently shaped wells for the capture plate for collection of metabolites on these larger wells, larger shaped wells. Again, all of these wells can be changed. Since this is all 3D printed, we can make any changes in design depending on the requirements of the project. So this is examples of brachypodium growth on plant growth on agar. As you can see, this is the plant root essentially grows on the membrane, and the metabolites are collected below the membrane in the capture plate. This is plant growth in soil. We can do root architecture imaging on using these. We can look at the fine root hairs as well. So we have optimized this platform for different types of plant growth, like, for example, brachypodium or sorghum growth in this case. We also have this humidity chamber setup to support the plant growth. So when we were optimizing this plant growth in soil, we noticed that it would dry up pretty fast. So we built this setup, and this humidity chamber can fit a lot of different of these 96 plate wells for plant growth. We can also, if a project requires, we can also create these SubTap rhizosphere platforms in larger formats, so that accommodates plant growth for longer periods of time. In this case, we have been able to grow a plant for up to 4 weeks. Okay, so again, like showing here, different the plant growth and the root architecture imaging, and we can look at metabolites from here spatially. So I'll show you an example how we have done that. And like I said, these platforms can actually be combined with bacteria growth, like in this case. So the experiment that we performed here was plant plus bacteria. So we had brachypodium growing in this well. We had Bacillus subtilis bacteria growing in the second well. And these two wells were separated by this membrane in the middle. Between well 1 and well 2, there was this window where there was a membrane fitted. So plant root and bacteria could communicate through different metabolites. So we did another repeat well of plant and bacteria. We had plant alone, bacteria alone, just soil, and then soil plus citric acid. To have a control, citric acid was basically a positive control, where we had like, we wanted to see that if we add like a synthetic compound, is it diffusing through the membrane? And so we captured all of this inside of these capture plate wells. And then this plate was sent to the mass spec for analysis. And so we get a whole bunch of data. As you can imagine, there's so many different wells that shows, that gives us, each well gives us like thousands of spectra. And we have workflow to analyze this data and represent it in a way that we can understand. For example, in this case, you are seeing like a PCA plot. Even though we see so many metabolites, the metabolites do cluster together. We do see root metabolites clustering together specifically. The blanks are basically soil or soil plus citric acid. We can cluster them separately. We do see a lot of root associated bacteria metabolite clustering in this area, and bacteria metabolites clustering a little separately. And then root plus bacteria with membrane, they alone, they are clustered separately. So we do see clusters of these metabolites, and as the project requires, we can go into depth of these analysis. All of these metabolites that we collect using mass spec gives us confirmation using tandem mass spec, so you can actually be confident of what you are observing, what kind of metabolite you're observing here. So spatially also we can kind of see some differences here, and you can see like this particular well where roots are observed, labeled A through H, and we see clustering of these metabolites. For example, the metabolites from the well A kind of clusters in this region, and then all of these different well metabolites clusters differently. And then once we start seeing these trends, we can then start using the MS or tandem mass spec data to identify what metabolites clusters where, and then try to develop like a biological story. What is the reason for this metabolites clustering together at that point? Perhaps there is some kind of signaling between plants and microbe, a plant root and microbe, or maybe in some cases there is some kind of deficiency. We can do different kinds of drought experiments because we can control humidity with our humidity chamber if the drought experiments is a goal in this case, for example. We haven't done more research into metabolites when plant and microbial metabolites in these platforms with just agar. We just looked at soil metabolites, but we can also incubate these plants in agar if you want to get a better idea or image of a plant root as the plant root is growing. Okay, so this is all I have today for all the two different platforms and what Jayde described the other two. So all the 4 different TerraForms platforms. As Jayde mentioned, all of these platforms are actually customizable. So these are only examples that we gave you. You could approach us to generate hypotheses, how we can test specific questions that you have in these platforms. Some of the things that are still being developed is how we can directly have like a XCT data and then create TerraForms in the future, how we can upscale certain phenomenon that we are seeing inside of TerraForms and do some certain kind of predictive modeling to inform us of events that can happen at core scale or even maybe ecosystem scales. So these are some of the upcoming technologies which we will probably talk about in the near future. Okay, now I think we can take questions from you guys.
All right, thank you, Arunima. Jayde, if you want to come back, we do have lots of questions. So I'm just going to start from the top. And actually, I feel like this is a good time to let everybody know that at AGU coming up in December, there will be a great giveaway at the EMSL booth. And there will be rhizochip and pore-scale micromodels available for visitors. So that's one good reason to swing by, and you can also chat with the staff scientists that will be there. So wanted to mention that. So first question comes from Tina. What is the significance of the rhizochip's oval shape? For example, why not circular or square?
Tina, we can definitely do circular or square. The oval shape for us, we were thinking it would maybe help guide the roots down the rhizochip and be less invasive than having to make the roots take like a 90-degree angle turn. But definitely customizable still. We can do different shapes as well.
Great. Andy Plymail has a question. In the synthetic soil aggregates, can the water saturation be varied?
Thanks, Andy. So it is a hydrogel, so it has to be hydrated with water. We can change the degree of methacrylation and the amount of gelatin methacrylate to water ratio to make, if you're interested in like changing the amount of water in there. But it is mostly like a saturated system.
Cameron Richardson's question, could you potentially grow multiple plants in one rhizochip?
I think you could, depending on how long you wanted to grow them for. It might just cut back on the amount of time that you could keep them alive in that small of a space. But I think there's no reason that you couldn't add another inlet and another plant if you're interested in looking at like plant root-root interactions or something like that.
All right. Amanda Stremecki is asking, can you inoculate rhizochips with mixed microbial cultures to simulate indigenous soil communities?
Yeah, totally. I mean, that would be really cool, I think, to look at more natural communities. It would limit the amount of, or the kind of imaging you could do, optical imaging, because none of those would be fluorescent, presumably. But if you were to look at maybe like the chemical imaging and comparing that to the spatial genetics, genomics, transcriptomics, then that would help you understand who is where and doing what.
Another question from Tina. For MPLEX, are there any barriers for bringing or sending soil from outside the United States for this integrated analysis?
That might be a question for more of our MPLEX team than me, but from what I understand, I mean, we do have international users, and as long as it's sort of like it meets USDA requirements, I think, and is not radioactive, we should be able to receive soils. It might just be a few extra paperwork hurdles to jump over.
She has a couple other questions. What is the pore-scale media for rhizochip? Is it air and water, like in soil, or only water, for example, one phase?
So the amount of volume in the rhizochip is about 250 microliters. You can saturate the system by adding that amount of liquid. You can also make it unsaturated by not adding as much liquid. I will say the plants uptake a lot of liquid, so you usually have to water them quite often. Unfortunately, we can't really say like we want this pore to be air and this pore to be water. Like we can change the total amount of water to air ratio, I think, by changing the humidity of the system. So yeah.
What about what is the pore-scale media for the micromodels? Similar to Tina's previous question, is there only air between posts? If yes, then where does the water come in?
So in this experiment, yes, it was air, because we tried to mimic a very drought-like situation inside of the pore-scale micromodels. So the carbon sources that we had in there had like agar in the carbon sources. And so what we observed is that the fungus moved the water into the pore space, and it remained there for very long periods of time, which is something that fungi does, translocate water, and within these soil pores, they can put the water in there for a very long time. But we can, for other experiments, we can add water media inside the pore space.
Question from Jack Kim. For the figure of synthetic soil aggregate versus liquid culture, how do you attribute the better growth in hydrogel to its aggregate structure instead of other non-soil properties, for example, absorptive properties unique to hydrogel?
I would say that we haven't quantified growth in the synthetic aggregate compared to liquid, so I wouldn't say it grows better per se or more in the synthetic aggregate. It just grows differently. It could be due to the pore space of the aggregate. It could be because it's a surface area that the microbes can grow on, and maybe they are activating other genes like biofilm genes or something. I do know the surface charge of gelatin methacrylate's negative, as is the surface charge of bacterial cells. So I don't expect it's like electrostatically sticking the cells to the surface of the gel, but that is something to dig into more, is understanding if it is a porosity thing. We might be able to change the porosity of the aggregate by changing the methacrylation of the gel, and that could maybe tell us whether or not it is strictly due to the pore size.
Nishant Paniker has a question. Is the plant microbe interaction analysis from MS1, is the QC decoder workflow publicly available?
So the data that I showed here, yes, it is MS1, but we do have MS2 data, and we can get MS2 data from these analysis. The QC decoder workflow is not publicly available yet, although for this particular analysis, we might make it publicly available. We are trying to make a more organized workflow for all of the wells that we have in the SubTap, so that users can just upload their data, and there's like a more automatic workflow available for the users as well.
Okay. Well, the next question is an important one. How do people access these resources?
Well, through EMSL proposals. And I think the next EMSL proposal cycle, which is the large scale cycles, these probably will open in December, January, which was what happened last year, around that time frame. And so you can contact us to better how to frame a hypothesis, or several different hypotheses that you have, and we can help you write these proposals. But through these proposals, you essentially request TerraForms.
Thank you. Larumia Faria's question is regarding the microchannel devices made with PDMS. How were the minerals incorporated into it?
So you can basically mix in with the PDMS, and the M Systems paper that we cited in our slides goes into specific details on how you can do that. But we don't use PDMS anymore for mineral devices. We use epoxies and other polymers. It's just easier. It's also it's also better compatible with mass spec.
Arunima, another question for you. Can you perform live in situ imaging on TerraForms platforms? Oops, you're on mute.
Sorry, not yet, but we're in the process of doing that.
Okay. And follow-up question, can you use the SubTap platforms as an incubation platform?
Yes, you could send us plant roots. And if you want to look at spatial metabolites, we can do that just using the plant root. It doesn't have to be growing the plant from the scratch. Okay.
Jade, question for you. Can you control where the microbes are added to the plant in the rhizochip to study chemotaxis towards the root, or whether or not the root will grow towards the microbial signal?
Yeah, so you can actually do that pretty easily. You just create another inlet on the rhizochip where you can insert the bacteria or fungus. We have tried that and added fungus to a different area of the rhizochip and actually saw the roots grow towards the fungus, which was surprising. So yeah, you can change where you're adding and inoculating the plant.
And for the synthetic aggregates, can you print a natural microbial community rather than a synthetic community of isolates?
Yeah, I think you can. That's something that we're trying to work on now to see if we can, for example, isolate maybe some different kinds of microbes from natural soil and just growing up a natural community in the synthetic aggregates. Most of the work we've done so far is working with isolates, but I think it should be possible to work with a more natural community, and that's something I would really love to work on.
Arunima, another question for you. What other experiments can be performed on the mineral TerraForms platforms? You talked about micro-mineral interactions in plant mineral interactions, but what other experiments?
So you can look at how the mineral chemistry is changing, how that particular mineral chemistry reacts with organic matter, and you can look at what kind of microbial necromass, for example, is attaching to which part of the micromodel, and that specifically happens on mineral surfaces. Those are some of the experiments. You could also look at how microbial hotspots are created when you generate a certain flow inside of these microchannels. We are also trying to make these platforms compatible with analyzing volatiles. So that's something super exciting, and keep an eye on, like, in the future, we will make announcement. So when that is developed, you could look at volatiles. The initial experiments show that it could be successful.
Jade, one more question for you. Are microbes able to swim through the bioprinted aggregate pores, or are the cells immobilized in place once you print them?
Yeah, we've done some optical imaging where you can see the microbes swimming around inside the hydrogel. We've tested the permeability of the synthetic aggregates, and it's pretty high, so things can diffuse in and out of there. Eventually the cells do sort of grow and start to consume the synthetic aggregate, and can start to grow outside of the aggregate itself too. So we are limited a little bit in how long you can keep the synthetic soil aggregates going. But initially I would say the microbes are mixed in, and then they can start to move around and escape over time.
Great. I'm not seeing any other questions, so I think we're going to wrap up for today, but I just wanted to give you one more piece of information. I mentioned the booth at AGU, that's booth number 515. So go ahead and write that down, and we hope to see you there. And then in January, we will be back with our webinar series. Hope you all have a great winter and holiday season. Thanks for joining us today.